Hello, biology students. This is Mr. Ward with a video lesson on carbon compounds as part of our Chapter 2, The Chemistry of Life. By the end of this video lesson, you should be able to summarize how large carbon molecules are synthesized and broken down, define monosaccharide, disaccharide, and polysaccharide, and discuss their significance to organisms, to relate the sequence of amino acids to the structure of proteins, relate the structure of lipids to their functions, and lastly, list two essential functions of nucleic acids. Key terms in this chapter you can find on page 54 in your textbook, which are carbohydrate, monomer, polymer, monosaccharide, lipid, nucleic acid, nucleotide, protein, and amino acid. So you will be creating a Quizlet for these key terms for your next class. When we talk about the chemistry of life, we focus on probably the most important element of the major elements found in life, and that, of course, is carbon. Carbon has its own kind of field of chemistry called organic chemistry. And when someone studies organic chemistry, they're studying carbon and the compounds that it creates. So compounds that contain carbon are considered organic compounds. And those carbon atoms are generally bonded to other carbon atoms and other elements such as hydrogen, oxygen, nitrogen. If you remember, 90% of all life is made up of the four elements: carbon, hydrogen, oxygen, nitrogen. That term CHON, as we often call it. And carbon has some very unique bonding properties. Remember that carbon is going to be sharing electrons. That's what covalent means. So it's going to be sharing electrons with other carbon atoms and with other elements. So because of that, they can create a wide variety of different types of compounds. And you can see some of the shapes that they take on in the images here on the screen on the left-hand side. So, for example, carbon can form a straight chain, and we call this an organic skeleton or a carbon skeleton. It can form a branched chain of carbons, or it can form a ring. Now, it might not quite look like a ring to you, but organic chemistry, we call this picture right here a ring of carbons. And when you see a single line between two atoms in these structural drawings, that means that they are sharing a single pair of electrons. If there's a double line between any of the atoms in a structural drawing, it means they're sharing two pairs of electrons. And if they obviously are sharing three pairs of electrons, that would be a triple line. So we refer to that as a single bond, a double bond, or a triple bond. And we see that a lot in these organic molecules, these carbon skeletons. So think about that as we move on to our next slide. Now, these organic skeletons, these carbon atoms bonded to each other and to other hydrogen atoms, for example, often we will find a cluster of atoms that join with or bond with that carbon skeleton. And those clusters of atoms often influence the properties of that carbon skeleton. So, for example, if we see that a carbon skeleton has an OH group attached to it, which is called the hydroxyl group, not to be confused with hydroxide, but oxygen and hydrogen combined together, because oxygen is a very electronegative atom, has a really strong pull on electrons, it gets a slight negative charge, and that also gives the hydrogen a slight positive charge, and therefore the hydroxyl group creates a polar molecule. So a bunch of carbon atoms all bonded together with a hydroxyl group will give it a slight charge and make it polar. The carbonyl group, which is a carbon double bonded to an oxygen, has some, also because of that, the nature of oxygen and carbon, it's going to have a slight charge. So a lot of times by adding a carbonyl group, it also makes that carbon skeleton a polar molecule. The carboxyl group, which is a carbon double bonded to an oxygen and also bonded to an oxygen, which is bound to a hydrogen, which would be COOH, when added to a carbon skeleton, will make that carbon skeleton more acidic. An amine group would be a NH2 group, which is nitrogen bonded to two hydrogens. And by attaching an amine group to a carbon skeleton, that gives it basic base property, so a basic alkaline property. And the sulfohydryl group is a sulfur bonded to an hydrogen. And one of the fairly unique properties of sulfohydryl groups they're fairly stinky. They give a bad odor. We'll just put it that way. That rotten egg smell when you're walking through a saltwater marsh and it smells, that's the sulfohydryl groups that are attached to the molecules in that substance they're being released. So these functional groups are important because they provide certain properties to the carbon skeleton that they are attached to. Now, moving on to macromolecules, and we're going to be talking about four major categories of macromolecules. What exactly is a macromolecule? Well, when we talk about macro, that term macro means large. So they're large molecules. And again, so think of that word molecule. It is atoms that are sharing electrons. They're covalently bonded, and that makes them considered molecules. So talking about large molecules put together. And when we look through the four categories of organic compounds in a few minutes, the term monomer is going to come up in at least three of the four. And think of it like Lego building pieces. A monomer is like a Lego building piece. So you're putting these Legos together or these monomers together, you can create a larger structure. That's what we see in three of the four macromolecules that are important to organic chemistry. And we see this picture here. These rings are meant to be carbon-based skeletons. It looks like we're adding them together to form a much larger macromolecule. So monomers are single building units, whereas polymers are a repeated linkage of these monomers together that form what's considered a macromolecule. So when does something become a big molecule? Generally, it's going to be two or maybe three or more monomers joined together. And they join together and they also are broken apart with a really important molecule that we've talked about. In something called a condensation reaction, we can join two monomers together by basically removing the water molecule. So, and we're not going to see that in this example, but in the next example, if we would like to break apart a larger macromolecule, we would add water to that molecule. So what would end up happening is the hydrogen of the hydrogen and the one hydrogen in the water molecule will be added, say, to this carbon right here, or maybe this oxygen, and then the OH group will be added to the other carbon that actually splits it apart. We call that hydrolysis. So a condensation reaction, often called a dehydration reaction or a synthesis reaction, is when monomers are joined together to form larger molecules. And the way we do that is we remove a water molecule from two monomers, and that allows them to join together. Just the opposite, we want to break apart a large macromolecule, a polymer. We have to add water to it. Just by adding a hydrogen to one part of a monomer and then the OH to the other monomer, that's going to cause them to break apart. We call that hydrolysis. That term lysis means to break. And one of the more important macromolecules for life is ATP, adenosine triphosphate, and it's a huge storage molecule for cells. And these phosphate groups, think about functional groups again, is a phosphorus atom surrounded by four oxygen atoms. By adding three of them together to a monomer of another class of organic compounds, that of course creates the ATP molecule. And in order to break a phosphate away, to give some of that energy to a cell for cellular work, we would simply just add a water molecule, and that would break that phosphate group away from adenosine triphosphate or ATP. So water, as we talked about in section 2, is a very important molecule, a very important compound, because it's involved in all sorts of chemical reactions. That's exactly what you see right here by either removing a water molecule from a substance or adding a water molecule. We can get it to either build new macromolecules or break them down. So water plays a very important role in the four organic compounds we're going to be talking about. So the first category of organic compounds that we'll be talking about is carbohydrates, which should be a familiar term, also known as sugars, also known as saccharides, and not all carbohydrates are the same. One of the characteristics of all carbohydrates is they have a general chemical formula of CH2O, meaning for every carbon and every oxygen, there are two hydrogen atoms in that structure. Now, the term monosaccharide, and again, saccharide refers to sugar, refers to carbohydrate. That term mono should mean something to you, a prefix meaning one, so it's a one sugar unit. And then that would again be the monomers of carbohydrates, or these saccharides, these monosaccharides. And you can see them, a short list here on the left-hand side, and some of them may be familiar to you: glucose, fructose, and galactose. So they have the same chemical formula, C6H12O6. However, they're put together slightly differently, which makes them isomers of each other. But glucose, fructose, and galactose are the building blocks of what can be larger sugars or larger saccharides, starting with disaccharides. So a disaccharide is when two monosaccharides are joined together. So this would be, again, an example of taking monomers and putting them together to form a larger molecule. Some of the more common disaccharides out there are maltose, which is a combination of two glucose monomers joined together. Lactose, which we find in milk, which is a combination of glucose and galactose. And then sucrose, which is often referred to as table sugar, is a combination of glucose and fructose together. So disaccharides are simply made up of two monomers, and there are a couple different types of those. And then as we get to the larger sugars, the large polysaccharides, think that prefix poly meaning many, many monomers linked together. And again, how do we get them? Well, we add the water molecule, and that allows the monomers to join together. And that's considered we extract a water molecule that's considered a condensation reaction, often called a dehydration reaction. Using that word dehydrate means to remove water from something. So some of the more important polysaccharides are starches. So for plants often store all of their glucose in starch, oftentimes in the roots of the plant for later use and maybe a different time of the year. There's another type of polysaccharide called cellulose, and that is a more structural polysaccharide found making up a majority of the cell wall of all plant cells. And then in animals, we find glycogen. And glycogen is our storage form of glucose. We store glycogen in our liver and our muscles. So when we need our blood sugar level drops, an easy way to raise that blood sugar level up is by releasing glucose that's stored in glycogen from either liver or muscle tissue. Another characteristic of carbohydrates is they have about 4 big calories or 4 kilocalories of energy per gram. And that'll become important when we start talking about our diet project. In the bottom picture here, you can see some good examples of carbohydrates. And a lot of times carbohydrates get a bad rap in different diets, but carbohydrates take the form of obviously fruits and vegetables, but we also have them in more human-created products such as sugars and candies, chocolate, of course, being one of them. But breads and grains and fruits and vegetables all are considered a good source of carbohydrates. And here are some diagrams, structural diagrams of carbohydrates. So for example, here is a glucose diagram for you. Now, again, I told you that glucose is made of 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. But you don't see them here. A lot of times in organic chemistry, when they're drawing the structures out, they don't often include the carbons. But we would see them at the intersection point of these lines, the corners of each of the ring here. So here would be carbon 1, 2, 3, 4, 5, and then outside the ring would be carbon 6. And then often some of the other hydrogens aren't shown as well. For example, this carbon would also have a hydrogen coming off of it in that direction. By combining glucose and fructose together, that would create sucrose. You can kind of see what that looks like right here. And then below is an example of starch, which is a long chain of glucose molecules, all forming at particular carbons in the actual structure there. Our next organic compound are lipids. Lipids as a group are considered large non-polar organic molecules. So if you remember that term, polar means slightly charged, non-polar means no charge. So therefore they do not dissolve in water. That's one of their properties. Lipids have a much higher ratio of hydrogens to carbons than to oxygen atoms. So for example, you could have a molecule, something along of C80, H120, O6. So the carbon to hydrogen ratio is much, much higher than to oxygen atoms. And that is a characteristic of lipids. So therefore, if you're given a chemical formula and it doesn't match that general CH2O, then you know it could possibly be a lipid. One of the examples of lipids are fatty acids, which are a long chain of carbon atoms bonded together, generally by single bonds. We'll see a picture of that, a diagram of that on the next page. So lipids don't have monomers, but we often see some repeated structures that kind of act like monomers, but are not truly considered monomers like we find in the carbohydrates. One of the characteristics of some of the lipids is that they have even though they are considered nonpolar, they do have some regions that are slightly attracted to water and other regions that are certainly repel water. This term, the philic part, phile means to love. In this case, so we have hydro meaning water. So in this case, the hydrophilic region is the water-loving region, so it's attracted water. Phobia, root word there, fear. Hydro, water, fears water, so it's repelled by water. So we do see some lipids that at one end they have a little hydrophilic region, and the rest of it is pretty much all hydrophobic. So we'll see saturated fatty acids in a moment here, but a fatty acid is simply a long chain of carbon atoms bonded together and then also bonded to hydrogens. When we see a saturated fatty acid, we would see a long series of carbon atoms bonded together with a single line in drawings, and then each carbon would be attached to two hydrogens in the middle of the chain. So you have a carbon that's sharing electrons on either side with another carbon atom and then two hydrogen atoms. So it's completely saturated, completely surrounded by hydrogen atoms, and it makes it again a nonpolar substance in general. Now, when we add a double bond to at least one between two of the carbons in the chain, then we have what's called an unsaturated fatty acid, and that slightly changes that chain from a straight chain to kind of a crooked chain, and structure leads to function. So this is perfect for performing certain functions. But one of the things we know is saturated fatty acids at room temperature are solids. Unsaturated fatty acids at room temperature are oils. So a lot of our plant-based oils we get are unsaturated fatty acids, and a lot of our animal fat sources are saturated fatty acids. Lipids as a group have twice as much energy per gram than carbohydrates. So roughly about 9 big calories or 9 kilocalories of energy per gram. This is kind of a short list of different types of lipids. So the word fat is kind of one little category of lipids. So we can't really refer to lipids are not fats, but fats are a type of lipid. So we have something called a triglyceride, which we'll see in a moment. Think of that word tri, 3. Fats, phospholipids. So we're going to add a phosphate group to a lipid component, and we're going to see that as the major part of the cell membrane. So a really important molecule. Steroids. So if we're talking about male steroids, female steroids, these are steroids that provide secondary sex characteristics. We also have cholesterol, which is a type of steroid. A lot of times steroids get a bad name in popular media, but they actually are some really important molecules that differentiate between males and females, and also provide some other really important characteristics to cell membranes. And waxes around an apple, for example, or the top of a leaf, an example of a lipid. Phospholipids, as I mentioned before, are essential components of the cell membrane. We'll be looking at those more in depth when we get to our cell biology unit. Testosterone and estrogen are types of steroids. So again, very, very important to the development of males and females. And as I mentioned before, are roughly around 9 to 9.5 calories of energy, big calories of energy, kilocalories of energy per gram. So lipids are a really important class of organic compounds due to the fact they can provide a lot of energy per gram for us. So even though, again, we don't really have monomers building lipids, we do see some repeating structures. So for example, this is the fatty acid chain I was mentioning, where you can see one carbon bond to another carbon in a long chain here. And notice that there are hydrogen atoms surrounding the carbon atoms. With the exception at the end right here, we do see a double bond, but when we get to the end of that fatty acid chain, that does not count. So this would be considered a saturated fatty acid. If we were to remove this hydrogen, carbon would need to form a double bond to this carbon here, and that would create an unsaturated fat. If we add multiple double bonds in the chain, we have a polyunsaturated fat, which are some of the healthier ones. In gray here, this is what's called the glycerol group, which is made up of 3 carbon atoms, made of 3 oxygen atoms, and made of, we have 3, 6, 7, 8 hydrogens. So that carbon to oxygen to hydrogen ratio is slightly different than in carbohydrates. But notice, in order to attach the glycerol group to the fatty acid chain to start building a lipid, we have to remove a hydrogen from this hydroxyl group, and then this OH from the carboxyl group from the fatty acid chain. That makes water, and now we can join them together. Notice down here we have 1 glycerol group attached to 3 fatty acid chains. This is a triglyceride, very important measuring tool to determine in terms of blood chemistry, what's our triglyceride levels. When triglyceride levels get too high, that could potentially lead to later cardiovascular disease. So this is an important lipid to measure in the bloodstream to keep, but it's almost related to our diets and a little bit of genetics as well. Our next category of organic compounds, not ones you would see on a food label, are nucleic acids. And nucleic acids also are made of monomers. We call them nucleotides. And you can see the nucleotide right here. We have the main portion, the sugar. So in DNA, it's called deoxyribose. In RNA, it's called ribose sugar. We have what's called a nitrogen base, and you can see that looks somewhat like a ring structure. Again, we don't see the carbons, but they are at every intersecting line. You can see in this particular case, there are 2 rings of carbons. And then we have a phosphate group, which is a phosphorus atom surrounded by 4 oxygen atoms. So that you see right there is the monomer of nucleic acids. So DNA would consist of 2 rows of these nucleotides joined together. And if you imagine that it's a ladder and you're climbing the ladder, the sides of the ladder would be repeating phosphate and sugars, and then the rungs of the ladder would actually step 1 would be the nitrogen bases. And there are 4 different nitrogen bases: adenine, thymine, guanine, cytosine. And in DNA and in RNA, we remove the thymine group and replace with something called uracil. And these nitrogen bases form those rungs of the ladder. So DNA consists of 2 rows of nucleotides bonded in the middle by the nitrogen bases, and you have a twisted appearance, almost like a helical staircase, and that's the shape of the DNA molecule, which is, of course, a type of nucleic acid. And RNA is just the opposite. It is also made of a long chain of nucleotides, but it's always a single strand of nucleotides, not a double strand, and they can take different shapes to form the different types of RNA molecules. But a very important molecule for life because it's the coded information of life. So we'll be talking a lot about the function of DNA and RNA as we get into our genetics section. And here we get into a little bit of a look at the nucleotide sequences here. So here we can see what looks to be kind of a spiraling structure right here. There's the different colors on the inside. Sometimes they're called nucleobases. These are the 4 or 5, depending if we're talking about DNA and RNA, types of nitrogen bases. So if we're talking about RNA, we have cytosine, guanine, adenine, and uracil. If we're talking about DNA, we have cytosine, guanine, adenine, and thymine. And you can see that these different colors, again, represent the different bases. So the bases join together in the center. The sides of this ladder are twisted into a helical shape in the case of DNA, which, again, is made up of true monomers, just like carbohydrates. And our last class of organic compounds, probably one of the more important ones, are proteins. And the reason I say this is because proteins have a wide variety of uses in any organism. Anything from structural to functional, meaning structural meaning muscle, made of protein, or functional enzymes that aid in chemical reactions. We'll be talking about section 2-4 are protein-based molecules. But what are these proteins made out of? Well, they are also made of monomers. We call these monomers amino acids. And here we have an example of an amino acid here. Every single amino acid starts out with a carbon atom in the center, a hydrogen atom bonded to that carbon, a carboxyl group, which again is a carbon bonded to 2 oxygens and a hydrogen in this form. And remember that carboxyl groups make the carbon skeletons that are attached to them slightly acidic. And then we have an amine group, which is a nitrogen with 2 hydrogens, NH2, and that amine group makes things slightly basic. So they kind of cancel each other out in a way in some cases. And then the R group. And what is that R group? Well, there are 20 different types of R groups, which mean there are 20 different types of amino acids. They only differ by whatever that R group is. It could be a hydrogen atom, or it could be a chain of carbons attached to that single central carbon. So it's different based on the 20 different types of amino acids. Just as an interesting side note, of those 20 amino acids, 12 of them are non-essential, 8 of them are considered essential. That means that even if you were to take in all of the right elements, your cells cannot actually build those 8 essential amino acids. You need to get them in your diet. Therefore, it's important to have a wide variety of foods in our diet with all different types of protein sources so that we get the right types of amino acids. And when 2 amino acids join together, it's going to be considered that bond between 2 amino acids called a peptide bond. So if we have two amino acids joined together, that's a dipeptide. It's another example of a condensation reaction. So by removing the water molecule, so in other words, removing a hydrogen from the amine group on one end of the amino acid, joining it to a completely different amino acid's carboxyl group, remove that OH, so that hydrogen and that OH form water. Now the two amino acids can join together, in this case, creating a dipeptide. When enough nucleotides are added together, we get a polypeptide, which is defined as a long chain of amino acids. And then eventually, when those polypeptides are joined together, a couple different types of polypeptides, we can that forms proteins. And the shape of the protein will often determine its function, which is very, very important. Shape determines function in biology. And proteins, like carbohydrates, have about 4 big calories of energy per gram, or 4 kilocalories of energy per gram. So as you can see, lipids have twice the amount of energy than carbohydrates or protein do. So when you talk about our diets, a diet that's high in lipids, high in fat, is going to have a high calorie amount versus a diet more of a carbohydrate or protein-based diet. Now, proteins can be broken down into various levels of structure. So the simplest way to start is if we string together a long chain of amino acids, that's considered a polypeptide. That's considered the primary structure of the eventual protein. Depending on some of the bonds that form from the hydrogen of one amino acid to some other slightly negatively charged part of another amino acid, begins to change the shape of that chain, gives it kind of almost like a folded kind of shape, or a little bit of a helical shape. That's considered the secondary structure. The tertiary structure is when some of these R groups that make those amino acids unique start to interact with other R groups and starts to bend that polypeptide even more. That's called the tertiary structure. And eventually, when 2 or more polypeptides come together and form a different type of shape, that's called the quaternary structure, the 4th level of structure in proteins. So those are the 4 types of organic acids, or actually organic compounds, that we'll be talking about a little bit more as we go through the rest of our year in biology. And of course, our next topic will be section 2.4. So look forward to seeing you guys in class the next time we meet.